Okay, 3, 2, 1, it's jam.
What's up, Science Fam? You may have heard people comment on your appearance and say, "Oh, you look just like your mom," or "You look just like your dad," or "Why don't you look anything like your parents? That's kind of weird." Well, looking like your parents is determined by something called genetics, and that's precisely our topic for this week. My name is Sir Ramon, and this is Science with Sir Ramon. Let's go! So, sometimes you get asked, "Kanino ka nagmana?" So, when you get asked this question, usually it's trying to pertain to who you look like, your mom or your dad. And basically, your parents tend to give you certain traits. You resemble your parents in certain ways. So your mom and dad, their traits kind of mix together and then it turns into you. But how does this happen? How do we end up with someone as unique as yourself? So our topic, once again, is heredity and genetics. It's how traits get passed on from one generation to the next. And so it's raining right now. You can hear the thunder, but who cares? So what's up for this lesson? This is what we're going to do. We're going to explain the laws of Mendelian genetics. We're going to see how offspring inherit alleles, whatever those are. We're going to find out in a bit. Compare Mendelian and non-Mendelian inheritance, and do some Punnett squares and compute for probability in genetics. All the good stuff. So last year, you should have already taken up a bit about Mendelian genetics. We're just going to have a review for this first video. So what is heredity? When we say something is hereditary, it means that there is a passing of traits from the parents to the offspring. So cute little babies tend to have traits from both of their parents. They may get their dad's eyes and then their mom's nose, stuff like that. But the question is, what exactly do parents pass on? We don't actually get anything from our parents in the case of body parts. Like if someone says, "Oh, you got your father's eyes," your father didn't rip out their eyeballs and give them to you. Okay? They pass on something else. What they pass on is what we call genes, this thing. So genes are sequences of DNA that code for specific characters or specific traits. So again, one gene codes for one trait. Now these genes are located on something called a chromosome. So chromosomes are, as defined, they're thread-like structures that are basically coiled up DNA. So when you coil up your very, very long DNA strands, they turn into chromosome molecules, and on those chromosome molecules are your genes. So the genes are what are passed on between parent and offspring. Now we get a set of genes from both our mom and our dad. So we have two sets of genes, two sets of chromosomes, one from daddy and one from mommy. So put them together from the sperm and the egg, we get what we call a diploid set of chromosomes. Diploid means there's two sets of chromosomes. Again, one from dad and one from mom. One from the sperm and one from the egg cell. So we say that the sperm and the egg cell are haploid, meaning they have half the number of chromosomes of a normal individual. So haploid means it has only one set of chromosomes from one parent. Moving on. Can genes be different? So we know that genes are on chromosomes and stuff like that, but how many different forms can one gene have? So alleles, this thing, an allele is a possible variation of a gene sequence, meaning it's a possible form. So one gene can have many alleles. Now, genes again are located on chromosomes. The physical location of a gene on the chromosome is called its locus. Now, for any one gene, it's always located on the same place in a chromosome, even from different people. So if you compare their chromosomes, it should be around the same place, like in this diagram here. This chromosome has it here, has that particular gene there, and this chromosome has that particular gene in the same physical space. So that's called the locus. Now, focusing on alleles, what are the possible variations of alleles? Usually there's only two allele types for every gene. Again, each gene has two allele types. We have the dominant allele and the recessive allele. Now, when we write this down in genetics, usually we signify dominant alleles with capital letters and recessive alleles with lowercase letters. So generally, the dominant alleles are overwhelming the effect of the recessive alleles. As you can see here with Deku and Bakugo. Bakugo in their childhood was the go-getter. He kind of just sinasapawan niya si Deku. Poor guy. What a loser. Anyway, so if we get two sets of genes, can the alleles from these two sets of genes mix and match? The answer is, of course, yes. They mix and match, and the way they mix and match can be defined as homozygous or heterozygous. So those are the two main categories. They basically mean for homo means same, so same alleles. In this case, we have two big B's in this one, so that means it's both dominant. We say homozygous dominant. If it's both lowercase, both recessive, we say homozygous recessive. On the other hand, heterozygous is one capital and one small letter, meaning one dominant and one recessive allele. Okay, so that's called heterozygous because it's different allele types. Now, can you have the same trait while having different genes? Can a person have can two different people have the same traits even though they have different genes for that trait? The answer is technically yes. So as we saw earlier, let's just get rid of my face. As we saw earlier, there's heterozygous, meaning a dominant and a recessive, and homozygous, meaning both the same allele. So in this case, if we have heterozygous and homozygous dominant, so both of these, big P, small p, and both big P, pp, they'll both code for a dominant trait. So that's a genotype. When we say big P and small p, or both big P, that's a genotype. Or the better way to say it is heterozygous or homozygous dominant. That's the genotype. It describes the alleles of the trait. On the other hand, if you're just observing the organism, that's called the phenotype, the physical characteristic. So again, genotype for the genes and phenotype for the physical appearance. So we can't tell what genotype it is basing on the physical appearance. It can be homozygous, dominant, or heterozygous, or perhaps even recessive. We'll find out how to find out later on. So just remember that genotype is for alleles and phenotype is for the physical traits. So now we move on to Mendelian genetics. Who's Mendel? Who made Mendelian genetics? So this is Gregor Mendel. Oh wow, look. What a typo. So he wanted to study, but he was kind of poor. So since he was poor, he joined the monastery to get a scholarship. So he got free education. And then he studied pea plants and somehow became the father of modern genetics. So lesson learned, be poor, get a scholarship, study plants, apparently. So Mendel found out three basic laws, three things about genetics that made him the father of modern genetics. The first one is related to this. Why are siblings different? They have the same parents, why not get the same traits? Why are you and your siblings different? So the reason is because of things like this. So Gregor Mendel, when he was experimenting on pea plants, he tried breeding peas and he noticed some weird stuff. So in this case, we have 100% yellow peas, and then their offspring included 25% green peas. How do you get green peas if all the parents are yellow? That doesn't make any sense unless you explain it in this way. So he figured that maybe, just maybe, what we see isn't what is. What we can observe isn't what is passed on in terms of heredity. So that means that there could be more than one hereditary unit, meaning more than one allele. So in this case, he figured out that the genotype can be different from the phenotype. Alright? And that's why there are instances where all yellow parents can make a green child. This is called the law of segregation. So each gene has two alleles. Again, each gene has two alleles. You can see it here. The parent can carry two alleles. Now, each allele segregates into different gametes. Again, when we say it has two alleles, each gene has two alleles, that's because it's a diploid organism. There's two sets of genes. Now, when those genes separate into the gametes or the sex cells, when you separate them into sperm cells, for example, sperm cells are haploid. So the diploid becomes haploid. You have to split them apart. So what happens is basically this. The big B goes to one sperm and the small b goes to another sperm. So these two different sperm cells carry different genetic codes for that trait. That's why siblings can be different, because one sperm can have different genes than the other sperm, and so on and so forth. Okay? So how do we figure out possible allele combinations? If different combinations of sperm and egg can have different allele combinations, can we calculate the chance of getting certain traits in our offspring? So we do that using what we call Punnett squares. So in Punnett squares, you have the top and the side. So these are the headers. So in the top part, or in the sides, we have the gametes. So again, each one is considered a gamete. That's why we draw like a little tail here. That means it's the from the sperm cell, from the father. So the father gives this kind of genetic code in that particular gamete. The mother, on the other hand, gives this type of genetic code in her egg cell. So if we combine the sperm and the egg, when we cross them together, you put in the square the combination of the genetic makeup in the gamete. So this capital A with this capital A will make two capital A's. You do that for every single box. Small a and big A, line them up to the proper box, we get big A, small a. And so on until we get the entire Punnett square. Now this Punnett square shows you the ratio and probability of your offspring. So there's one box that has double capital A in 4 boxes. So there's 1 out of 4 chance that you'll get a homozygous dominant offspring, meaning both capital A. Now for heterozygous, meaning capital A and small a, you have 2 boxes out of 4. So 2 out of 4, lowest terms, 1/2, you have 50% chance to get a heterozygous offspring from this combination. And you have 1/4 chance of homozygous recessive because there's only 1 box out of all 4 boxes that's recessive. Alright? So why are these 3 colored? It's because heterozygous genotype will still have a dominant phenotype. Again, the dominant allele will overwhelm or mask the effects of the recessive allele. So this small a doesn't matter when it comes to phenotype. Again, the physical trait, the physical characteristic. So it will look the same as this thing. Again, the traits of homozygous dominant and heterozygous are the same dominant trait. So only homozygous recessive will have recessive traits. That's why these 3 are the same color, because they will express the same trait. So 75% dominant and 25% recessive in terms of phenotype. So again, genotype and phenotype can have different ratios. Now there are special Punnett squares. There are special Punnett squares like the monohybrid cross, which means 1 trait. Mono means 1 and hybrid means a cross, or a Punnett square. Dihybrid, on the other hand, crosses 2 traits, which we'll discuss later on. So again, monohybrid, just 1 gene. Dihybrid is 2 genes. Okay. Now another special Punnett square is the test cross. I won't explain that because that's going to be part of your brainstorm topic. But this image shows you the basics of a test cross. So I'll let you guys discover that on your brainstorm discussion. So can one gene affect another gene, or genes inherited in sets of genes, or are they inherited as is? So the answer is that we'll find out. So if what happens is that they're inherited as a set, meaning they're clumped together. So you have, for example, the gene for color, which is symbolized by the letter Y, and the gene for shape, symbolized by the letter R. If they're inherited in sets, what happens is this. So each box is inherited as one thing. So in this case, we have a yellow round P and a green wrinkled P. So if you cross them together and they're inherited in sets, your phenotypes should be just these 2. So you started out with 2 phenotypes, you should end with 2 phenotypes, which is the round yellow and the green wrinkle. Because, again, they're inherited as a set. Okay? But if they're not inherited as a set, this is what would happen. Let's get rid of my face. So there were more combinations than expected. There's 4 different possible combos. There's round yellow, round green, wrinkled yellow, and wrinkled green. And that's because the alleles of different genes were distributed. Meaning, they weren't inherited as sets. The alleles just got segregated, got separated, and then distributed to different gametes. What does that mean? We have the law of independent assortment. So if we're dealing with 2 genes, for example, each gene has 2 alleles that are distributed independently. What does that mean? Getting rid of my face, we can see this diagram here. So we have capital Y and small y. We have to distribute those 2 alleles to 4 different sperms. So this capital Y gets distributed to these 2, while this small y gets distributed to these other 2 sperm cells. Now your capital R and small r, a different gene, has to be distributed in the same way. You distribute capital R to 2 sperm and your small r to another 2 different sperm. And you get 4 different combinations. We have capital Y, capital R, capital Y, small y, capital R, and so on. So these are different combinations of alleles on single sperm cells. So this sperm cell is carrying capital Y and capital R. This one is capital Y and small r. And because of that, we can conclude that your alleles are independently assorting from one another, meaning your Y gene doesn't get distributed in sets with the R gene. They do things their own way. They don't affect each other. A simpler way to think about it, if you like math, is the FOIL method. In the FOIL method, if you have binomial expressions that you have to multiply to one another, like x plus y times x minus y, you distribute them based on first, outer, inner, and last. And that's basically what's happening here. The first ones, capital Y and capital R. Boom. Capital Y, capital R. The outer ones, small y and small r. Boom. Small y and small r. The inner ones, small y, capital R. That's this one. And the last one, the outer ones are capital Y and small r, and so on. So again, using a FOIL method, you can get the possible gamete combinations. Okay, so don't forget your math. It's useful in biology as well. Okay, so again, this is independent assortment. 2 different genes don't affect one another. They don't get inherited in sets. Alright, so the last law we already talked about is the law of dominance. Again, a dominant allele will mask a recessive allele. So we're going to take a break because that's a lot to absorb. So we're going to continue in the next video. I'll see you guys.
Party's over.